Hello, everyone. This is Raj Thakur. I'll be talking about ocular drug delivery, which is a major area of research interest in my team. So now, before I proceed discussing about the drug delivery, I'd probably like to give you a bit of introduction about myself. So I'm Professor Raj Thakur. I'm professor in pharmaceutics. I'm also the chair in pharmaceutics. I was earlier a research associate in the same department. I also have a number of different roles outside the university, such as the member of editorial board for the ophthalmic drug delivery and frontiers in drug delivery. I'm also an assistant editor in ophthalmic or ocular focus group in drug delivery translation research. Previously, I was vice chair and chair for Ocular Focus Group at CRS. You can probably find a bit more about myself at the link you could see in this slide. So I think that's really my brief update about myself. Now, obviously, the lecture is on ocular drug delivery. My team does work on ocular drug delivery. We're also exploring some other areas and solving some of the chronic disorders. So this lecture is primarily focused on ocular drug delivery and the technologies that we're working on. Now, before I get into talk about technology, let's understand the problem with the eye and the seriousness of the issues across a number of eye diseases. So as you could see on this particular slide, visual impairment is a significant health and economic challenge. Here in the UK and worldwide. There is a rising concern about a number of people suffering with disorders of the eye. Visually impaired people around the globe or in the world is about 285 million, of which around 39 million people are blind, and around 256 million have some degree of visual impairment. 65% of people with visually impaired and 82% of all the blind are 50 years and older. So I think that really defines, as we get old, we tend to have more issues with the eye, and probably do so with the rising number of even with the young generation using computers and phones and other gadgets. I think probably this COVID has really restricted us more at home, and so we're more onto these devices for several hours in a day. So this does have a lot of implications apart from our changing social and food habits. So just looking at this particular slide, this one here, you could see every 5 seconds someone in the world goes blind. And obviously that's a serious concern. And here in the UK, developed nation, we see every day around 250 people in the UK start to lose their vision or sight. I think a part of the problem is this here, as you could see on this image, there are a number of different issues with the eye, both the front of the eye or back of the eye. So you can see on the top row, there are diseases which are primarily seen in the front of the eye, such as, for example, blepharitis, conjunctivitis, probably you might have experienced at some stage. Uveitis, dry eye syndrome is another major and big problem, particularly in the people using contact lenses, and also it is one of the common ocular disorders. And obviously, something more slightly serious would be cataract. There is a huge number of operations of cataracts done globally, that is replacing the intraocular lens. So it's a well-known problem, and there's a well-known solution to treat it. But if you look at slightly more serious issues with the eye, let me use a marker here. Okay, that's fine. So diabetic retinopathy, you're talking about age-related macular degeneration and glaucoma. I think these three are much more serious in terms of treating for a long term. As you could see, the nice and normal vision of these two boys holding the balls here can really get distorted with the diabetic retinopathy patient and age-related macular degeneration. Their loss of central vision, and glaucoma is a slow and irreversible disorder. So the seriousness of the eye diseases, front and back of the eye, need to be dealt with better technologies in treating these disorders. And there are a lot of complications associated with that I'm going to talk in next few slides. So first thing first, eye on this image of this slide is what we get to see. So you have your iris, sclera, a part of sclera that is visible, and which is where we normally use eye drops. But the bigger challenge is the back of the eye, which is where we need medicine, particularly treating the back of the eye disorder. So when it comes to really when we're trying to look for a solution, whether you have conjunctivitis, you're looking for treatment more front of the eye. So you're looking here in the aqueous humor, where you are trying to deliver your drug, or top of the eye, which is your key target. But if the disease is more at the back of the eye, you're more kind of looking to deliver drug in choroidal area, retinal area, and more kind of a vitreous area to treat the disorder such as AMD or diabetic retinopathy. So then your target is really at the back of the eye, which is obviously invisible from the front, so it has to be injected. I will talk through why is the case. And likewise, if you look at more kind of glaucoma, your target can be even iris or ciliary body. We're trying to reduce the intraocular pressure. So I think it's really important for us to understand what disease we're dealing with, and accordingly, your key target tissues are different across a range of ocular disorders, and therefore you could actually design an appropriate formulation or a technology, a device that could help to treat some of these disorders. So this is a nice, wonderful diagram drawn by my student Mohammed Faris Adrianto. You can see the paper recently published about talking about various different in vitro models for measuring release of drug from an ocular formulation. Anyhow, so if you look at this particular structure of the eye, it's very complicated, as you could see. For example, let's take the anterior segment. As I said, anterior segment diseases such as blepharitis, conjunctivitis, dry eye, etc. drug need to be administered on the top of the eye. We are very familiar with where we administer eye drops or different formulations. So part of the challenge is that every tissue involved in the front of the eye and likewise at the back of the eye does have a varied degree of complexity. For example, even we're looking at the simple tear film itself, they have several layers of it. You have a lipid layer on the top, you have an aqueous layer, you have a mucus layer, and then your corneal epithelium. And then if you look at the cornea, then you obviously have, here you could see, you have corneal epithelium, and then you have Bowman's membrane, and then you have stroma, and then you have endothelium. So it's more kind of a lipid, hydrophilic, and hydrophobic barriers. So these barriers does limit the amount of the drug that could cross across these barriers. So it's more kind of a static barrier, but at the same time you have a dynamic barrier, you have metabolic barrier, you have enzymes that degrade the drug, you further reducing the bioavailability. You have blood flow that will carry away the drug from the site of administration. For example, when you inject drug subconjunctivally, a lot of the drug is lost to the other areas of body, including systemic circulation. So there are a lot of these barriers, but I think if you go to the back of the eye, the problem gets even more complicated. It's complicated enough, even a healthy tissue, a healthy eye, but the complication gets even, it gets very challenging even in treating a disease eye where the complications are less well understood. For example, if you're looking at delivering the drug to the back of the eye, you're more or less targeting in this yellow region, which is retina. There are several different layers within the retina. You have the white tissue of the eye, the sclera, then you have choroid, they have suprachoroidal, then retinal layers, and then you have obviously the blood vessels within these tissues that also limit the amount of the drug that could reach into this particular very thin and very complicated structure. One of the other problem is also you have vitreous humor, which is a jelly-like material that fills your eyeball. As very complicated features. It's not just a gel of hyaluronic acid of around 1% sitting in water of 99% and a few other components, but it's a lot more complicated than that. It changes in viscosity over time. You have a lot of other problems such as syneresis, etc., etc. I'm not going to talk about those challenges at this point, but what I really want you to understand in this particular slide is the segments. You can divide the eye into two segments: posterior segment or anterior segment. You can probably need to look at what kind of disease we're dealing with. Then we have different barriers to understand. And then you have to effectively design a delivery system to overcome some of these three key elements, which is a static barrier, metabolic barrier, and the dynamic barriers. So these barriers does reduce our bioavailability of the drug. So then comes, and obviously what we have learned about diseases, we have learned about the barrier functions, the three barriers. Then what options do we really have in terms of delivering drugs to the eye? So as you could see, the anterior segment, one kind of formulation that probably everybody is very familiar with is the eye drops. They're very well known. They're very simple. We have either taken it or we have seen people taking it. So it is most commonly used medicine or formulation strategy for treating ocular disorders. But there are some other approaches. For example, slightly less invasive. For example, the subconjunctival injection, which is given just behind the conjunctiva, outside the eye still. There are other methods or areas of administration of the drug. For example, subtenon, periburbar or retiburbar. So these are the injections given more on the outside of the eye. I think probably the most invasive or most commonly used method, yet most invasive, is the intravitreal route. As I said in the previous slide, because of these barrier properties, because eye is an organ which is not easy to access, particularly the back of the eye, one way of getting the maximum amount of the drug in the eyeball to treat the back of the eye disorders such as AMD, diabetic retinopathy, is by injecting the medicine directly into the eyeball, and which is commonly called as intravitreal injections. There are other less researched or less commercially driven technologies such as intrascleral has been tried. I'm going to talk about it, but that's another potential route to deliver medicine within the tissue. In this case, it's the white tissue, which is sclera. So we have various options of routes of drug delivery. We know there are barriers function. We know the disease origination, either front or the back of the eye. So the engineering of this formulation, when you deliver at this location, it is what really we're looking to solve some of the unmet needs. So I thought maybe I'll just talk about some of the challenges with topical drug delivery. Topical drug delivery, as I said earlier, is number one route of delivering drugs to the eye. Eye drops to treat front of the eye disorders or anterior segment disorders. Now, eye drops themselves have several challenges. The moment we put an eye drop, you're either going to blink or rub your hands on top of the surface of the eye. Or there are certain elements or there are certain excipients such as preservatives present inside the formulation that could trigger your tears, which will actually further drain the eye drops and remove the drug that is being delivered. So reducing further the bioavailability. Eye drops that we put on to the eye typically around 20, maybe even 40 microliters, but the eye can really accommodate around 7 microliters. So not surprisingly, because of all these actions here, blinking, tear drainage, and then rubbing the eye, this would all lead, and the barrier function, which is static barrier, metabolic barrier, and then your dynamic barrier, will further reduce the absorption of the drug, and therefore most likely it's 5% or less than 5% of the drug is absorbed. Poor compliance, particularly we think it's an easy process to take an eye drops. The amount of the force that we apply on the eye bottle, depending upon the design of the eye bottle, can vary, and that force can change that amount of the eye drops that falls onto the eye surface. And again, the natural process of blinking and rubbing your eyes, etc., further drains the drug away. So there's a significant issues with the compliance. 80% of the glaucoma patients fail to administer the drops on regular basis, and 90% of those patients do not really apply eye drops appropriately, and some of them have challenges even pressing a bottle. So as we get old, we have a dexterity changes, and that have implications. When you try to add more than one eye drop, for example, post-cataract treatment, the patients are given different eye drops to treat the eye. The compliance even drops because the patients have to deal with more than one drop, and any problem you see with one just simply multiplies. So it also depends upon your properties of the molecule. Molecule cannot be too large in size, ideally 500 Daltons or even below, for it to show some level of permeation across the cornea or conjunctiva. And then we also talk about how you make the formulation, the solubility, what kind of solvent you use, the pH, the charge can also significantly affect the permeation of the drug. One way of probably retaining more drug for slightly longer period of time is, instead of giving solution, you can also have suspensions. But again, you're limited in terms of the size of the suspension, and ideally you want to keep it 10 micrometers or below. So there are significant issues. For example, this image here, this is use of a prostaglandin eye drops. Because a lot of the drug land on the outside of the eye, you can see that the darkening of eyelids does happen. And this is a medicine, something that has to be taken. The prostaglandins, such as latonprost, has to be taken on regular basis for several years together. So so these are some of the very typical challenges with eye drops. And obviously, we have to come up with some solutions. This is an image to show a quick solution, and not an ideal way of doing it, but there is a significant desire to have a better way of delivering eye drops if I have to keep it short, in terms of overcoming some of these challenges. Right now, if you look at topical delivery, eye drops, we're familiar. The other option you have, if not topical, is probably periocular. Or we can also say transscleral injections. Sometimes we can even say intrascleral injections, depending upon the injection area. Subconjunctival injections are very well known. Less commonly known is peribulbar, subtenon, or retrobulbar injections, which are given outside the eye. So this really is a factor. What is really happening in here is you administer the drug outside this white tissue of the eye, and you let the drug to permeate across the tissue, either to the front of the eye, if that's a target, or back of the eye, if that's a target. For example, retina, if that's a target for the back of the eye. But one of the problem really with this kind of delivery system is, obviously, drug is not going to stay in the spot you inject. Obviously, it's going to move. If there's a circulation happening, there's a dilution happening, it's entering into systemic circulation, which is very well being proved. But then the problem really is depending upon the size of the molecule. You cannot expect to have a large biologic molecule, 40, 50, 60, 100,000 Daltons molecule to simply walk across this white tissue, the sclera. So there is a lot of physicochemical properties of the drug molecules that you need to consider, take into consideration. including the polarity, including the charge, not only the size, but also the hydrodynamic radii. That is also important for the effective permeation of the drug. And now there's a number of different research papers that have been published that do talk about various different other ways of enhancing this permeation by using a device-based system, for example, iontophoresis and other kind of devices. I will talk about a few of them. Even upon these injections, it is not necessary that you will see a huge rise in the bioavailability of that particular drug. There's a great systemic clearance that does happen before you get effective levels of the drug for a long period of time to the target tissues, such as the retina or the aqueous humor for treating the aqueous glaucoma. So then comes the slightly more invasive way of delivering drugs to the eye, is injecting straight into the eyeball. As you could see, these images are not great, but this is something patient has to go through every month, every 4 weeks, every 8 weeks for more or less the rest of their life in order to save their vision. So these are called the intravitreal injections. Intravitreal injections introduced way back in 1911. Not that highly used from then to around 97. You could see there were very little number of injections happen until maybe, I would say, maybe 2006, 2007. But 2006, 2007, around that area when the anti-VEGF drugs came in, the number of injections really skyrocketed. So you could see around 2001, you have less than 5,000 injections, which has went straight close to 1 million injections by end of 2007. So what's really happening here is now these intravitreal injections or injections directly into the eye has become a very gold standard therapy in treating diseases of the back of the eye that I showed in my earlier slide, where because these diseases are treated with biologics, and because biologics are big molecules, you can see, let me see. Yeah, you can see the size of this small molecule versus the large molecules. It's massive. It's a big size. Not only size, the physical chemical properties, etc., does not simply allow them to pass from the topical application with an eye drop to the back of the eye. Therefore, you need an injection. An injection is now clinically practiced for several years for injecting some of the medicines to the back of the eye. For example, bevacizumab, ranibizumab, or aflibercept, the commercial products Avastin, Lucentis, Eylea, are injected into the eye between 4 to 8 weeks period to treat, at least to maintain the vision of these patients suffering from AMD or diabetic retinopathy. One of the biggest problem is these proteins have a high cost. The barrier is a big issue, therefore you need injection. They are short-lived or short half-life, and therefore you need to inject them more often, and it's like poking the eye again and again and again. So, and there is a stability issue with this biologics. Again, I'm not talking much about the biologics, the challenges here. This lecture is more to give an overview of what we do at Queen's, and particularly my team, what are they working on. So there, normally I do talk about the issues, particularly with biologic. But I think in general, the intravitreal injections, which are given into the eye, as you could see on the image, are very invasive. Has to be given every 4 to 8 weeks. This is a poor drug distribution. Or maybe I can even say we understand little about the drug distribution, especially in the disease eye. There's challenges in actually measuring that. And then there is a short half-life of the drug, and there's a poor patient compliance. So obviously, you can imagine getting injection into the skin is scary. Getting injection in the eye is not surprisingly very scary. So patients actually stay away from getting injections. Some patients are really scared of getting injections in the eye. However, you could see on this particular graph, there's a significantly growing number of injections at exponential rate. of getting these medicines in the eye in order to at least maintain the vision. So there were roughly around 23 million injections given globally of these anti-VEGF biologics or drugs. And another thing is become a very routine practice. So probably one way to get away from having routine eye drops or injections in the eye is to have a sustained release formulation or long-acting system or controlled release formulation. So you all might be familiar with the peak and troughs effect of regular intake of medicine, such as probably this is more conventional picture used in oral administration. I'm trying to depict the similar scenario in case of intravitreal injection. So as I said earlier, so obviously injections are painful. They're they're definitely not comfortable. So you need certain eye drops before and after treatment to control the pain or discomfort. There are less chances, but there are chances of endophthalmitis and also rise in intraocular pressure. There's a chance of retinal detachment, not to mention the number of other complications. So one way of getting over these problems is trying to probably inject a formulation that perhaps lasts for a long period of time. So at least that will reduce the need for frequent injections. So again, this schematic really trying to represent one injection probably sufficient for running up to probably 6 months in the best case scenario, or even more. To treat the back of the eye disorders. So obviously there's a major clinical and commercial opportunities exist for developing some novel sustained release drug delivery systems. Okay, so I'm going to just really quickly talk through this particular slide. There's a reference for this very nice figure. Probably I missed to add in here. But I think this is a nice figure to actually talk about what everybody else are trying to do and what are different approaches they're trying to look in order to overcome some of the challenges that I was discussing earlier. So there is obviously improved in terms of devices, use of topical application, passive diffusion device. There's a endophoresis device to enhance the permeation of the drug. Then I also talked about transscleral or intrascleral delivery, injections of materials within the tissue of the eye, use unidirectional implants, use of hollow microneedle, dissolving microneedle. You talk about injecting implants inside the eye via intravitreal injection, or even trying to suture some of the non-degradable implants that could probably release the drug over a long period of time to overcome some of these problems. Exactly the text on the left-hand side, really trying to reiterate some of the information that I just shared earlier. So there's a critical need in order to overcome or in order to address some of the unmet needs in treating AMD, treating diabetic retinopathy, treating glaucoma, and other parts of the eye. So let's look at what has been done. So okay, we know there is a problem with the eye. We know there are 3 big barriers. We're looking at static barrier. We have metabolic barrier. We do have a dynamic barrier. Then we have diseases of the front of the eye. We have back of the eye. So we have all these problems. We know these are problems for many years. But if you look at what has been done, we also know that sustained release can help, which is great. But look at this particular, again, another wonderful diagram also drawn by my PhD student, Faris. So here you can see what has been done to date on addressing some long-acting system or sustained release system for treating ocular disorders. Now, way back, Alza developed probably the first device or implant for the front of the eye in 1974, which was a week's release implant delivering pilocarpine in treating glaucoma, called as Ocular. One of the challenges with this particular device was the implant was falling off in certain patients, and the patients were not comfortable. So there were some clinical issues, and therefore the product has really dropped on the market. And not until probably around 22 years after, we had another long-acting system, which is wet reset. Wet reset, red reset, both are based on the same principle, similar technologies, delivering different drugs. One is delivering ganciclovir in treating CMV retinitis. The other one delivering a steroid, fluocinolone estenoid, for treating the posterior uveitis disorder. And then came in 2009 a 6 months long-acting and probably the first biodegradable implant to the back of the eye, treating the DME, diabetic macular edema. The product is Ozurdex, contained dexamethasone, launched in 2009. So which was great because a degradable material does not need an additional surgery to be removed, whereas the wet reset does need surgery to implant and also to remove it from the eye, which probably can be negated by using a degradable material like this one here, Ozurdex. So 2011, Iluvan came in, which also delivered the same drug that Retisert delivers, and then Utic and Iluvan are pretty much the same product, but indicated for different indication. So again, that also contains the same drug. Drista was then the second degradable implant treating glaucoma, which is injected in the front of the eye in the aqueous humor, a small implant which is also made from PLGA, which is a material also used in making Ozurdex, launched by Allergan. And just very recently, a non-degradable device has been approved for delivering by biologic, which is ranibizumab by Genentech. So again, you need a surgery to put in an implant, but this is first device which is actually delivering biologic, unlike the rest of the devices which are focused on delivering small molecules for varying periods of time. So that's the reason I was saying it's more kind of a 6 implants really, because Retisert are similar type of implants, uses different drug. Iluvan, Utic are exactly same, but used for different indication. Ozurdex and Drista, both are based on PLGA, hot melt extrusion based technology. So in reality, we have around 6 implants developed over the course of 47 years. Now, probably I'll leave that discussion for another kind of lecture where I can actually normally talk about why it is not easy to come up with delivering and making implants in solving some of the disorders in the eye. So there are huge challenges. It's not a simple task in order to develop devices or sustained release systems for the eye, because eye is much complicated, much complex, I must say, in order to develop a device that successfully can be translated for commercial usage. So obviously you can see there's a growing interest particularly in ocular drug delivery since the onset of the anti-VEGF and proteins to the back of the eye. So you could see between 1995 to 2005, there are only around 3,500 hits if you type that in Google Scholar. But between 2006, which is around the same time when the intravitreal injections usage has risen, until 2020, you can see that has significantly increased in terms of research and R&D activities. So obviously there's a growing interest in this particular area. I think, if I'm not wrong, don't hold me for what I'm saying, but I think ophthalmology is probably the second leading area of interest, revenue, etc. After cancer. So there are good drugs. Good drugs do need innovative solutions in order to overcome some of the challenges that I just spoke. So I want to keep this particular slide very brief. We're working on various different systems. I can't accommodate some of them here. I probably We have some IP work going on. But we have in our lab at Queen's, we're working on various different formulation. We're working on long-acting injectable gels that form depot upon injection. We're also working on preformed injectable implants for the treating back and front of the eye, delivering small molecules, large molecules. We're also working on minimally invasive dissolving hollow microneedles. We're also working on particle-based systems that includes microparticles, nanoparticles, microfluidic fabricated particles. We're also working on micelles using tri-block diblock copolymers. We're also working on 3D printing in order to produce implants, particularly addressing some of the ocular conditions. We are probably the first group ever who have actually used laser-based microporation of ocular tissues in order to demonstrate the clinical advantage of delivering drugs in post-laser therapy. And use of laser is well known in ocular drug delivery. So in addition to working on drug delivery platforms, we're also working on medical devices. What I really mean by that is we are working on devices that enable us to do more kind of a precision injections of medicines in the eye. So a part of our group is also involved in synthesizing both polymers and peptides and therapeutics. So we do make them in-house. There are projects that produce materials in order to address some of the diseases or challenges in the eye and even beyond. We're also working on self-assembly peptides or hydrogels, hydrogels conjugated with small molecules on delivering drugs to the eye. We're also trying to overcome some of the challenges with topical drug delivery, like eye drops, and delivering a better formulations using electrospun materials. So we're working in various different areas in addressing some of the challenges in my lab. I'm going to talk very quickly through some of the published work. I'm not talking much about what we are currently doing, obviously, because of the IP issues. So here, a preformed implant, this particular project was more on fabricating implants using PGDA, polyethylene glycol diacrylate polymer, where we photocross-link it. So you could see that we could sustain some of the small molecules. Here we have used triamcinolone estanoid as one of the small molecule. You could see we can sustain release the drug with less burst release going up to probably 250 days and then slightly over 250 days with a single implant of around 10 by 5 mm in dimension. So obviously we also look at number of different properties of these implants. You could probably refer to this article for much more details of what we have done. So we all one of the key factor with the materials, as I mentioned earlier, that you want them to biodegrade. So look at the degradation. One way of characterizing them at looking at the microscopic image of these implants, how the structure changes over time, particularly the porosity of the material, and so on. So we do measure them using number of different parameters, number of different techniques, and one of them obviously is scanning electron microscopy here. So you could see that the drug release could be controlled, could be tailored by changing the polymer concentration, and then looking at more dimensional effect. We're looking also at the composition of the polymer, the drug loading, etc., etc. So this is one way of trying to sustain release drug molecules. In this case, we have actually, you could see the right-hand side, we can sustain release proteins, as we have done in this particular paper, where we have used ovalbumin as our model protein, which is very close in molecular weight to that of ranibizumab, commercially called as Lucentis. There's another project that we have done where we have looked at depot forming ability of the phase inversion implants that uses PLGA and N-methylpyridine as a solvent. And obviously you might be familiar, we have a review on phase inversion materials in JCR. I probably have not put it in here, but you could again see that. So these particular images that you see This was the first ever method of non-invasively looking at the implant formation and degradation using OCD I was just thinking what is OCD stands for. So it's coherence topography, so optical coherence topography. That's it, I got it. Yeah. So it's OCD allows us to look at the thickness of the implant that form at the outer edge and how that implant gets harder over time and how the implant degrades. So we did some work. There's another paper coming from the group that actually thoroughly characterizes and by non-invasive ways of looking at the implant formation and degradation over time. So you could see the implant formation and implant decreases in its dimensions over time, literally indicated that the material that we have used in this particular paper, which is PLGA, allows us to sustain release triamcinolone for over around 2 months or even beyond. That could be tailored by changing in the composition, the volume, the size, and loading of the drug. The other interest which we are largely driven with is trying to use the not so conventional ways, not topical, not interpreted, but something intermediate, which is more looking into intrascleral or transscleral route of delivering systems that sustain release a drug over a long period of time. So that's another way of doing it. So we are currently investigating a number of different type of formulations. The whole idea here really is trying to get the drug very close to the choroidal or retinal area, so that you get enough drug levels and maintain those levels for a longer period of time in order to sustain release the drug molecules. So one way that we have done, as I said earlier, this is probably the first ever study that has been done using the fractional laser from the police device, which has largely been tested in enhancing drug delivery across the eye, across the skin. We have the first time actually used for the eye, and we have looked into a number of different parameters. So this particular laser device uses iridium YAG laser. So it's a erbium doped yttrium aluminum garnet laser. Wow, I got that right. Not an easy word to pronounce. So this laser does really help to generate the pores, and there's a different mechanism that it does. So use of laser is not uncommon in ophthalmology. It has been used to, especially in ablation of tissues, diseased tissues. So at a very short wavelength that is strongly absorbed by the tissue proteins, and probably the mid-infrared wavelength is strongly absorbed by water. So mid-infrared and excimer lasers have been clinically used in ocular surgeries. So one of the advantages of the use of mid-infrared lasers over excimer laser is are they less or non-carcinogenic and non-mutagenic. I think that's one of the advantages. And particularly the use of these YAG laser comes at the wavelength comes around at the absorption of water. So this allows us to absorb a lot of water, which then creates the pores. So we have actually shown, you can see that before laser and post laser, you can see the pores created within the tissue. Sclera is a complex tissue because the orientation of the fibers is really different as you move from the front of the eye to the back of the eye. So it's not a consistent structure, and particularly the strength of these tissues or the fibers orientation does change over time, and it gets weaker obviously as we grow old, as eye gets old. So we did show that using various different laser settings, we could create pores, less or more, and using different energies, we could actually selectively generate pores to certain depths within the tissue, not going beyond the whole length of sclera. But you could particularly see in here, before and after laser, the rhodamine is a small molecule. You could see the accumulation of the drug is much higher with the poration. Likewise, if you see the FITC dextran, 150,000 Daltons is a huge molecule, which is very close in molecular weight to that of Avastin or bevacizumab. You could see there is a more green patch you could see compared to non-laser or before laser application. We also looked at the tissue recovery. I wouldn't really say we have looked into very great detail, but I think this is a very typical way of looking at transcranial water loss kind of measurements. However, the tissue recovery is probably different in vivo. This is more kind of an ex vivo test or in vitro test. So at least we know that there's a certain degree of recovery happening. I think it really depends upon the depth and the density of these pores that we create. So there's another work that we did with the intratranscranial delivery systems using microneedles. I'm not going to talk too much about these microneedles. We have our colleague, Professor Donnelly, who is going to talk, done great deal of work on microneedles. So here, what we're really trying to do here is trying to use two of the strategies that are typically used in microneedle application to the skin. So in this case, microneedle, you can apply solid poke and patch technique where you apply the needle, take it out, put a patch, which might work pretty well with the skin. The other one is really coat and poke, which is we coat the drug, ideally waxing onto microneedles, apply to the tissue and remove it. You're leaving behind potent medicine in the tissue that is enough to create the therapeutic benefit, just like waxing. So in here, we're particularly looking at soluble microneedles, which are more kind of can be used for ocular application, and importantly, hollow microneedles that allows you to deliver medicine into the tissues very selectively to a selective depth. So that's the reason why we had done a lot of work using soluble needles and hollow microneedles application to the eye. So there are a number of different publications we have done. These are just an example of what we have done. But you can probably go through these publications to see the extent of work that we have done, in particularly with the ocular application of the microneedles in treating some of the front or the back of the eye disorders. So again, you can create molds. For example, you can see here a mold containing a conical shape opening where which was used to pour the gel in order to make the microneedle. You can see on the right hand side. So there's a process in how we make it. So we can laser engineer the mold, which is PDMS, a silicon mold, where we can create the pores of defined dimension, the base diameter, the height of the needle. You can define all those parameters. And then simply you add the choice of the gel that is probably more compatible with ocular tissues, and then spin it down in the centrifuge, you dry it, and then you take the patch out, and then you will find the needles. And we have worked on different sizes, and obviously we can change that. So idea really here is, how much force does it require to push this needle into the tissues? That is very important character to know. How much time does it really take for the material to dissolve? And what really happens to the material that is deposited in the tissue, and what happened to the tissue, whether it recovers, how fast it recovers. All these are the typical things that we have investigated. We have, for example, looked into transcript formation of albumin, again a model protein. You can see here there's a great degree of accumulation of a 70,000 Dalton FITC dextran into the tissue before and after. Likewise, with the 150,000 Daltons, there's a slight increase in the permeation or deposition of these particular big molecules in the tissue. And you can also load microparticles. We have just shown in the recent publication, we can load microparticles that could be deposited in the tissue, which can act as a long-acting system. We have also done a lot of work on injecting materials into the tissue. In fact, there's a paper certainly more advanced in terms of materials that we have been working on. So here really is, instead of cutting the tissue and putting a tablet inside the sclera, which has been reported on several literature, what we thought really is to inject materials into the tissue itself, which by using the hollow microneedle, you can deposit the materials. It could be a thermoresponsive material, in this particular paper, we have actually used poloxamer. We are all aware of polyoxamers that could be deposited within the tissue, and over time, the tissue encapsulates and form the material then forms a depot within the tissue, which then can release a drug which is loaded in it. So we have actually started making polymers specifically addressing some of these diseases and enhance the release period of the drug for a long period of time. So we have actually started designing materials to specifically address these particular minimally invasive long-acting system for delivering small and large molecules to the eye. Probably talk a little bit more about, so in addition to leading my own group on developing new technologies and new solutions for treating ocular diseases, in the process of which I've also found a spin-out company. So I'm the founder and chief technology officer at my spin-out called Rivana Therapeutics. So Rivana is a preclinical stage company that looking into commercializing some of the novel long-acting systems that perhaps overcome some of the typical challenges we have seen with some of the long-acting systems which are in the market, or some which are at an early stage of investigation. So in here, as you could see, we are making a raw step implant. You could see here this particular image really showing ultra thin wall needle loaded with this particular implant of ours called as iLeaf, which is a pre-formed implant that could be injected. The mechanism really is there's a slow swelling of the material and then the release of the drug over time. One of the neat thing about this particular implant is it allows surface erosion that has a significant advantages over bulk erosion. So we do a lot of polymer characterization and testing in order to thoroughly look into these kind of long-acting systems. It does come with a lot of challenges that we have actually addressed. So you could see the release of a biologic molecule here for almost 6 months. So in a way, the challenges that I was talking about earlier, a single injection of a biodegradable implant injected into the eye can sustain the release of some of the biologics for really long period of time, so 4 to 6 months. We can adjust the release kinetics of the drug. We can deliver hydrophilic small molecule, hydrophobic small molecule in addition to biologics. So, and I think we have done a neat engineering allowing us to deliver biologics. We have filed several patents, particularly for these drug delivery systems. The other commercial product that we're interested in developing in Rivana is Ocular, which is a depot-forming long-acting drug delivery system. So it does allow us to inject small amount of material, a gel, into the eye or other areas in the body where we could sustain release proteins for very long period of time. Here you could see we can sustain release protein for over 12 months. I can't talk too much about the materials here, but yeah, you can follow us on Rivana, and you could see how we're trying to address some of the challenges in overcoming developing a long-acting system, particularly addressing the disorders of the eye. So I think this is a great progress we have doing in addressing these challenges. I think I finally like to thank my team. We now call it as the GODS team. That really means the great ocular drug delivery systems team. You can see all the wonderful people here. My present PhD postdocs on the right-hand side in the lab in all the white coats, and there are some of the other past students on the left-hand side. And you can see the wonderful image of the eye. That's a huge image. That's not photoshopped. It was on display in Belfast in Ulster Museum for some reason, but we thought we'd take an opportunity, walk to the park and take that image because that's a brilliant moment to capture. With that, I would like to thank everybody listening to this talk, and to reach out to us. You can connect to us on our lab Twitter handle, which is Thakur Lab _KUB, and obviously you can connect to me as well, or you can drop an email to me. I'll be happy to talk. So thank you very much. With that, I'll probably close this lecture.